This video will be covering the basic steps that many next-generational sequencing methods use. First, we will go over what next-generational sequencing is, and some of the common names that it is referred to as. Next-generation sequencing is DNA sequencing methods that involve chemical assays other than the traditional Sanger deoxy chain termination method, which is also referred to as the first-generation sequencing. The Sanger method is a very long and work-extensive method of sequencing, and the next-generation sequencing is much faster and is able to separate more than one strand of DNA at a time. Other names for next-generation sequencing is deep sequencing or massively parallel sequencing. It can also be split up into second and third-generation sequencing by whether or not the DNA being sequenced needs to be amplified or not. In second-generation sequencing, the DNA undergoes amplification, or production of thousands of copies of the template molecule, by either PCR or bridge amplification. Third-generation sequencing is also referred to as single-molecule sequencing, because it does not need to be amplified and sequences the single DNA molecule of interest. The basic steps are library preparation, clonal amplification if it is second-generation sequencing, and then the sequencing itself. After the sequencing is finished, the data must be processed and analyzed as well. The library is a collection of DNA fragments that is stored and propagated into a population of microorganisms through the process of molecular cloning. In order to prepare the library, it must first be fragmented into smaller pieces of about 300 base pairs that can be sequenced. The process of cutting up a large strand of DNA, sequencing it, and then putting it back together is referred to as shotgun sequencing. There are various ways to fragment DNA. One common method used is sonification, which shears DNA by exposing it to periods of high sound energy. Another common method is nebulization, which shears DNA by forcing it through a small hole in the nebulizer unit. This results in the formation of a mist that is collected. The fragment size is determined by the pressure of the gas used to push the DNA through the nebulizer. After fragmentation, the ends of the obtained DNA fragments are repaired and an A overhang is added at the 3' end of each strand. Afterwards, adapters, which are necessary for amplification and sequencing, are ligated to both ends of the DNA fragments. These fragments are then sized, selected, and purified. Next is clonal amplification, which can be accomplished in two ways. One is by PCR. In emulsion PCR, they use an oil and water-based method, where one emulsion bead is attached to a single DNA fragment, and then the fragment is amplified. Each bead has its own fragment. The other method is bridge amplification, which uses the solid surface flow cell-based method. This creates clusters in which each cluster is made up of many of the same DNA fragment sequence. Each of these amplification methods are discussed in greater detail in other available videos. There are various methods of sequencing, such as pyrosequencing, sequencing by ligation, and reverse dye terminators. Pyrosequencing works with sequence incorporation of nucleotides and using luminescence. With sequencing by ligation, they introduce oligonucleotide probes with fluorescence. And reverse dye terminators incorporate reversible dye terminators that use fluorescence. Mapping is one of the many ways that the data needs to be analyzed after it's been sequenced. There's also visualization and quantification. All of these are done using bioinformatics tools. This is the works cited for this video. In the next set of videos, we will be going over various sequencing methods and how each work in greater detail. The first will be the first generational sequencing method of Sanger dideoxy, and the rest will follow will be the various next generational sequencing methods.